The process of gene expression occurs in two steps. In these two steps, we go from the information encoded in DNA, or our genotype, and turn it into the information encoded in a protein, or our phenotype. In this process, we go through transcription, or going from DNA to RNA, and then we use the information in RNA in the process of translation to produce a protein. So let's look at an overview of this process of gene expression. So the genes in the DNA are sort of the instruction booklet for building an organism. So your book likes to talk about this process as a cookbook, or the instructions for building a meal or building a particular dish. So since your book uses cooking instructions, I'm going to use an instruction manual just to give you a different way to think about it. And really, it's really similar. So in this instruction booklet, in this example here, we're going to build a protein called crystalline. So crystalline is a protein that is present in your eye. It's a protein that absorbs light and helps form the image in your brain of what you see. So here we have the crystalline gene. This is a specific locus on one of our chromosomes, the crystalline gene. If we look at the DNA sequence here, we have a short sequence of it. The process of transcription, that first step in going from DNA to RNA, is going to make a copy of one side of the DNA. That copy of one strand of DNA is going to be translated into another molecule called amino acids. That amino acid chain is then going to be folded into a functional protein. So we're going to talk about this two-step process of transcription and translation in order to produce a protein. And we're not really going to get into how proteins fold because we didn't get into the chemistry of proteins. That was chapter 3 and we skipped that, and that's okay. So I'm not going to talk about how protein folding actually happens. Just keep in mind that once this amino acid chain is made, it has to be folded before it can become the functional protein that has a job within the cell. So, transcription and translation, the two main steps in gene expression. In transcription, a copy of a gene's sequence is made. This is a slightly different process depending on whether it's a bacterial prokaryotic cell or a eukaryotic cell. In the process of transcription, DNA is made into RNA and then can immediately be turned into a protein, amino acid chain. This can also be called a polypeptide. So it's a pretty simple process in prokaryotes. In eukaryotes, we have a couple different steps. Because we have a nucleus, we have to first change our DNA into mRNA, messenger RNA. This is the molecule that is going to code for just a small piece of our gene. That mRNA has to be processed so that it can move out of the nucleus to the ribosome, which is our organelle that's going to produce our polypeptide chain or our amino acid chain. Polypeptide is just another word for amino acid chain. So the process of translation then is the translating from our RNA molecule into a new language of amino acids. So here is our book's analogy of how the process of transcription works. We can take our piece of DNA, identify a gene that we would like, open up that DNA double helix, make a messenger RNA copy. That messenger RNA copy is processed and moved out of the cell to the ribosome, where the ribosome constructs our protein molecule. So our book likens this to using your grandmother's cookbook. So the cookbook is the entire set of instructions for all of your grandmother's cooking, but you just want to make chocolate chip cookies. So in messenger, the analogous process to transcription, you would transcribe the recipe onto an index card. You would take out just the chunk that you need and take that to your baking counter in the kitchen, where during the process of translation, you take all of those ingredients and combine them into what will eventually become your functional molecule or your chocolate chip cookie. In my analogy with a instruction manual, you would take just the chapter, just the set of instructions that you want for building, let's say it's a Lego castle, and you just want the section for building a tower. So you are going to, probably not, but for my analogy, you're going to rip it out and you're going to take it to the desk where you're going to build it. And then in the process of translation, you're going to go from the printed information in your Lego manual to the physical construction of the tower on your castle. And then when you're ready for the next part, you'll go back to your manual, rip out the page, if you do that sort of thing, bring it to your desk. I realize this is not foolproof because I personally don't rip out Lego pages, but you know, trying to make it work here. So anyway, that's how the process works in general, if you want to think of it using an analogy. On Launchpad, as part of our homework this week, you will find animations for all of this. I strongly suggest that you watch them. You actually get credit for it, so go do that. Because all of these processes are complex processes with moving parts, and they're much easier to understand when you can see the moving parts and what's actually being built. All right, so part one, transcription. In transcription, we are going to take the information from DNA and turn it into RNA. In transcription, we make a copy of just the gene that we want to use to make the protein. So this, of course, requires that source, the DNA. It requires a large molecule called an RNA polymerase. Polymer means a chain of molecules. Ase means we have built them. So RNA polymerase is a molecule that is going to build a chain of RNA. To build that chain, we need the components, which are RNA nucleotides. And in the end, we are going to produce a long single-stranded RNA molecule called messenger RNA. And we call it messenger RNA because it takes the message from DNA in the nucleus out to the cytoplasm at the ribosomes, where that message is going to be transcribed. So we're going to use DNA as our template for this, and we're going to make an RNA molecule. So transcription happens in 4 steps. Let's talk about the first 2. In the first step, called initiation, we start with our DNA sequence. Now, one of these sequences is going to be called the template strand. That's the one that we're going to use to make our messenger RNA. This is all going to start at an area called the promoter. So this is a sequence of DNA that a variety of large molecules recognize as the beginning of a gene. So all genes have this promoter region. They actually all have this little box called a Tata box, which is a recognizable sequence for those proteins or other molecules. Within the promoter region is the transcription start site. So on your study guide, this is labeled as TSS. This is the actual specific base that transcription is going to start. So that is a really important, it's like the signpost that says, all right, everybody's in place, we're ready to go, now start here. Here is our big RNA polymerase molecule. So once all the other necessary ingredients are there at your DNA template strand, your RNA polymerase, this large bubble-shaped molecule here, is going to bind to your DNA, and it's going to open up that double helix, sort of like a zipper at that start site. So remember, I mentioned very briefly hydrogen bonding. Those are the bonds that bind the base pairs together. And I said that individually they are weak, but together they were strong. So these hydrogen bonds are holding the sugar phosphate backbones together. But when RNA polymerase comes in, it breaks just a few of those bonds and starts just a little bubble where RNA polymerase can access that start site. Remember I said that A's and T's bind with only 2 hydrogen bonds? That's why this Tata box here has A's and T's. There are less hydrogen bonds to break, and so it's easier to pull the strand apart. So that's why RNA polymerase binds at that location. So initiation is the process of all the pieces coming together so that RNA polymerase can bind at that promoter region and open up the double helix to use it as a template. So this is like you opening up your instruction manual or your cookbook. And then you find the specific chapter or recipe that you need. In the second step, we have transcription. Now, this is also called elongation. I learned it as elongation, so I might accidentally refer to it. I'll try to use the term transcribe because that's what your book does. In this process, the RNA polymerase will start at that transcription start site and begin adding complementary base pairs. Now here we have the messenger RNA hanging off the end. So this has already been added to complementary base pairs within the template strand. And now we are continuing along. See, A binds with T, U binds with A, C binds with G. Now this just moves along like a train on its track. Now you might be thinking that I just screwed something up here. I said A binds with U. So one of the big differences in RNA compared to DNA is the presence of a fifth base called uracil. In messenger RNA and another RNA molecule we're going to talk about in just a minute, actually all RNA molecules, this uracil base is utilized instead of thymine. So when you make a messenger RNA, the complement to adenine is actually uracil. So this single-stranded molecule is made of A, C, G, and U instead of T. But it otherwise is complementary in all the same ways that DNA is complementary. So you can see how we are using one of these strands as a template. Now that is a key feature of DNA's double helix structure. So we have bound our promoter region. RNA polymerase is chugging along, transcribing the section of the gene that we need. So this image here is a little bit more complicated, and it's got all 4 steps, but I want to just focus on this recognizing and binding. So our RNA polymerase has recognized a region here, this green region here. It has bound and has begun adding RNA nucleotides. So here we can see the RNA polymerase adding in nucleotides that are complementary to the template strand. As it moves along, the messenger RNA sort of hangs down like a tail. And as I said, this is kind of like a train running along a track, and as it does, it's adding new sections of track. So the second half of transcription is the termination and processing steps. So in the termination step, RNA polymerase moves along the DNA template, making its messenger RNA until it reaches a specific termination sequence. Now this termination sequence sort of jams up the RNA polymerase, causes it to stop, and the whole system falls apart. The DNA double helix goes back together. RNA polymerase can go bind somewhere else and make another messenger RNA. And then that messenger RNA is ready to go. It's all done. If it is a prokaryotic cell, it will just go right on over to the ribosome where it's going to be turned immediately into a protein. If it's in a eukaryotic cell, that messenger RNA has to be processed. And the reason that it has to be processed is it has to be stabilized so that it can be moved from the nucleus where the DNA is housed out to the cytoplasm where the ribosomes are. Remember from chapter 4 that those ribosomes are studded in the side of the rough endoplasmic reticulum. So we have to move this messenger RNA from the nucleus out to the endoplasmic reticulum where the process of protein synthesis is actually going to occur on those ribosomes. So the first thing we have to do is we have to cap the ends. So one end, we add a cap. The other end, we add a tail. Now this is a diagram from another book that is much more complicated than you guys need to know. But I like it because it shows some of the important features that your book doesn't. This 5' cap and this tail are going to stabilize the molecule. They're going to act like little beacons that say, hey, pick me up, I'm ready to go. And then the other end is going to say, hey, don't eat me, I need to be used in the cell. Because your cell doesn't want these messenger RNA molecules running around. Once they're used up, they're going to break it down. So by adding this cap and tail, it protects the ends of your messenger RNA so that it can be used to make a functional protein. Within here is our start site where transcription started. So I'm sorry, ignore what I just said. That's something completely different. Not all of it, just the part where I said start site. The other part of this last step in transcription is editing. So if you were going to turn in a paper, you would check to make sure the title's good. You would check to make sure you have your conclusion paragraph. And you would check to make sure there's nothing unnecessary in the middle. So in the editing process, those introns, those non-coding regions that we talked about earlier in the lecture, are actually taken out, and the remaining region, the coding region of your gene, is stuck together. And so you end up with this process messenger RNA with the cap on one end, a tail on the other, and just your coding region on the inside. It is this processed messenger RNA that's going to go into translation as its next step. So here we've got our termination site. So this RNA polymerase is going to move along our DNA template, making our messenger RNA until it gets to this termination site here. That termination site is going to signal for the RNA polymerase to release, and your messenger RNA is going to float off. Your molecule is going to be capped and processed. So we add our cap down here, we add our tail down here, and the non-coding regions, the introns, are removed, and we are left with our mature messenger RNA that can be translated into a protein. So the cool thing about this whole process is that the RNA polymerase essentially unzips the DNA molecule. It unwinds the double helix. And as RNA polymerase moves off of it, it actually rewinds and zips back together because of complementary base pairing. So once your RNA polymerase is finished with the DNA, it just sort of whoop, right back together and can go back to hanging out in the nucleus until it needs to be used next time.